The first topic today is the dark side of science, how you can find things that both clinicians and scientists can help prevent. So we're starting with the story.
Story time. This is a story about the Demon core, which is a plutonium core. This was back in the 1940s when they were conducting some essentially early nuclear research. So, a scientist by the name of Daglian was stacking tungsten bricks around this core, and each brick that he added to this brick enclosure was bringing the core closer to critical mass. While attempting to stack another brick around the assembly, Daglian accidentally dropped it onto the core and thereby caused the core to go well into supercriticality, a self-sustaining critical chain reaction. He quickly moved the brick off the assembly but received a fatal dose of radiation. He died 25 days later. So, hindsight is 2020. We would now look back and say, why on earth would you be stacking these reflective bricks around a plutonium core? Because we understand more about the consequences. But one of those areas, the dark side of science. Interestingly, this isn't the only accident that this core was involved in. I believe there was a second incident before it was melted down and repurposed.
That's pretty scary. In healthcare, we have this Swiss cheese model where there's multiple stops. So the medical assistant is looking for a stop. Perhaps the MSL is looking for a stop as well. And then the healthcare provider is sort of the rind in the Swiss cheese model of healthcare. It's definitely scary to be working with something that can literally kill you, like radiation poisoning. But it's also scary to be working with patients where you can literally kill them as well.
Yeah, I think when I was working in the hospital, it's a great example of how essentially all of us would be functioning within that model. You have the scientists who are doing the research, testing the safety, testing these treatments. You have the physicians that are ordering these treatments for patients. You have the pharmacists that are verifying there's no drug interactions. And then you have myself, an RN at the time, who is scanning things into the electronic health record, yet another layer, making sure that there's no interactions there, no contraindications with the patient's current clinical condition. So to have a bad outcome, really so many things have to go wrong, and it's much better that way. More layers of safety.
You know what, it makes me think of a lot of testing that was done and a lot of studies in anabolic steroids with drugs like fluoxymesterone in the 60s, 50s, 60s, 70s. That now we would definitely not use those drugs, but back then they were used and trialed for recovery and anabolism. And that was very interesting because now we know better about how they are harsh on the liver, all the 17AA anabolic steroids. And we would see that now thinking, oh my God, there's layers of safety that must be implemented before giving someone 50 milligrams of Halo, right? So that's just some of the things that we've seen. I think even recently with the ban on the over-the-counter Superdrol, right, or Epistane, or all those drugs that we're familiar with now, and we're thinking, but didn't it that cause a lot of liver issue, a lot of liver damage, and you go to the local GNC and you get something that is really harsh on your liver. How does that happen? And I think now we have obviously much more experience. So we are more cautious. It's not as fun. You don't really see studies on fluoxymesterone or whatever, DMZ or whatever, but yeah, you don't cause as much damage.
It raises an interesting question of how do you arrive to these conclusions, and the logical approach has generally been animal models.
Right.
And we have talked about in the past the low clinical translation rate with using smaller animal models. For example, murine and rat models have approximately a 2% clinical translation rate. So it's always interesting to see which bodies of researchers and clinicians either support or oppose using animal models to elucidate some of these drug interactions, and carry out these studies. So then the logical question is, if not on humans, what is suitable? Is in vitro studies sufficient? As computational chemistry gets better, people are starting to use a lot more computational drug-ligand, drug-drug interactions. So I'm curious what you guys think about this.
Maybe you can clarify a bit, but intuitively I would think, okay, you have a promising preclinical finding maybe in a mouse model. So you think, okay, we'll see, does this translate to human tissue in vitro? So if you have positive findings there, then you say, okay, what about human tissue in a living organism? So you have human cell lines in a mouse model. And then that's when you would progress either upward to human trials, depending on the safety, or to larger mammalian models. But I'm sure you understand the process much better than me. I'm just speculating there.
Yeah, that's generally the order of operations. And it's interesting to see which animal models are preferred. In other countries, you often see canine models used, horse models used, but in the U.S. generally, I think, at least in my experience, we strove to hit pig models, porcine models. So it's, it also, back to the dark side of science, brings up, in the past, we learned a lot doing ape experiments and monkey experiments. And for example, Morgan Island off the coast of South Carolina now has a population of 3,500 rhesus monkeys, of which approximately 600 are harvested for studies each year by the NIH and NIAID. So there's always there's pretty consistent pushback on doing that, but at the end of the day, if we're not using high fidelity animal models for these tests, humans are really the last experiment, and so there's a lot of unanswered questions if you remove that animal model to begin with.
It seems like as you go up in size, like nobody has problems with experimenting on fruit flies or nematodes. Some people are like, oh, well, the mice, maybe that's a line that some people are not comfortable with crossing. They say, no way. Go to a canine model, people will say, absolutely, no way, can't do that. Go to a primate model, and people are like, no, that's too sophisticated of an organism, no way. But then you eliminate all those, you're really escalating the potential harm each time. If you go right to testing on humans, very bad things could happen.
Exactly.
It depends on the potential benefit as well. So are you running a trial on an animal that is purely to see what happens or for toxicity or whatnot? Or are you running a trial that could potentially be therapeutic as well? For example, in the dog aging project, they give dogs that are prone to certain types of genetically linked cancers due to genetic bottlenecking during the world wars. And they give them a drug that's rapamycin, mTOR inhibitor, and it certainly has the potential to help. We know that in mouse models it helps, and it's well tolerated even in human studies. So a project like that, a lot of individuals who are against other forms of canine clinical trials would be okay with that.
Yeah, giving the potential for your man's best friend, your pets, your loved ones to live longer, healthier lives.
Yeah.
So you have to look at the potential benefit, and in general, I don't know the results of that trial haven't been released yet, but I would suspect there would be a low incidence of adverse outcomes, and at worst a neutral effect, and at best a positive effect based on they're really the stepping stones that have been studied on the other clinical models, preclinical models.
Yeah. Speaking specifically about dogs, it's also interesting to see what outside of clinical trials culture or society is willing to accept. For example, they're willing to accept neutering or removing the testes of dogs. That's no big deal at all. There is some studies on vasectomies for dogs to maintain their quality of life and decrease the incidence of fractures later in life, so on and so forth, and sarcopenia. But that's totally fine. But then if you do a clinical trial that has a well-tolerated pharmaceutical potential and then relatively low incidence of side effects and the potential for benefit for both canines and humans, then that becomes not okay.